So yeah, so I guess we're ready to start. Can I just go up again just to if you just stop sharing for a minute.
Do you want me to stop sharing?
Yeah, I'll just say hello to everybody in Portuguese.
Yeah.
Bom, gente, são 4 horas, bom dia, vou mudar de língua, I'll switch languages for a bit, mas enfim, bem-vindos ao nosso webinário da Iniciativa Brasileira de Reprodutibilidade. Obrigado a quem está aí no Zoom ou via Facebook, aos nossos colaboradores e ao pessoal de fora. A gente tem algumas notícias, a gente está terminando os protocolos da iniciativa, provavelmente até o final do mês. A gente finalmente conseguiu que todo mundo revisasse em tempo aí e tal, para quem está a par do nosso escopo de prazos aí e tal. Mas, enfim, a gente tem acho que 1 ou 2 laboratórios devendo revisões ainda, mas a gente está consolidando isso tudo, espero que a gente tenha isso até o final do mês. A gente está começando a fazer compras, a gente acabou de encomendar células, etc. Então, assim, as coisas estão se mexendo. Provavelmente no mercado de predição deve acontecer em novembro, a gente está terminando de passar pelo comitê de ética agora, assim, a gente vai anunciar isso publicamente, uma vez que os experimentos estiverem abertos, que o mercado estiver aberto. E a gente vai estar, realmente gostaria que todo mundo participasse, qualquer pessoa com experiência em pesquisa biomédica, em pesquisa experimental em qualquer área, na verdade, pode participar, a gente pode falar mais disso depois. So now I'll switch to English to present Dr. Nathalie Percie of Sert. She's head of experimental design at the NC3Rs, which is an institution in the United Kingdom, which is meant to improve animal research. I mean, mostly with the 3 Rs, or refinement, replacement, and
reduction. Reduction.
In terms of like using less, but also, I mean, having better, I mean, improving animal ethics also involves doing better experiments, doing more solid experiments. This is what the Brazilian Reproducibility Initiative is all about. We have around, out of 60 experiments on which we're starting, I think like 25 or a little less than half are involve rodents. So, I mean, we definitely have a big issue on performing the best animal experiments possible. And I think Nathalie will talk to us about some tools that they have been developing. Nathalie has been the first author of the revised Arrive guidelines. You might have heard of those. If not, I mean, you should have heard of those. And well, I'll leave it open for her to talk what the, and there's all kinds of free online resources in STVR. So I mean, I think most of us who work with animals can actually make use of those. So thank you, Nathalie, for accepting the invitation. And
all right, no problem. Thank you for the introduction. So let me try to share my slides again. Can you see my slides? Mm-hmm. All right. So yeah. All right. So so my talk is in 2 parts. You mentioned that you wanted 20 minutes and then question. So basically, in the first part, I'm just going to talk about the background, why we're doing this, and kind of like, just give general information about subjective bias, randomization, and blinding, why they're important, and also sample size determination. And then in the second part, I'll talk in more detail about the resources that we've got at the Iniciativa and how you can use them when you're designing or conducting or reporting animal experiments. So just in terms of, just to start and just give you a little bit of information about where we're coming from. So I'm from the NC3R, so that's the National Centre for the 3Rs in the UK. So we're an independent scientific organization. We've been running for 15 years now. And basically what we do is that we use the 3Rs, so replacement, reduction, refinement, to support better science and to support innovation and animal welfare. So we have 38 staff, mostly postdoc, based in London. And we've been running a program of work on experimental design since the NCCR started. And the reason that we're doing this is because if animal experiments are not designed to yield robust results, if they're not reported in enough detail, then they actually can't be used, and that's a complete waste of animals. So that's really the biggest waste of animal that you can think of, and that's why we've got this program on experimental design, to try to address this waste and improve things. And in the UK, animal experimentation is authorized based on a harm-benefit analysis, and I'm sure that's the same in Brazil. It's the same in the US as well. So when you do this, you really look at the benefits to science, the benefit to society, and you weigh that against the likely harm to the animals involved. And this is where experimental design and reporting comes into play, because if you don't, if the experiment is not designed to yield robust results, if people can't actually use the results, if people can't build on the results, then actually the benefits are just not realized. And that means that this harm benefits analysis is just not realized, and there's just no benefit to the science. It's completely unethical to the experiment in the first place. Sorry, I've got an issue with my slides. So when we started that program of work, we actually started by looking at the quality of published animal research. So this was 15 years ago. And we sampled about 300 publications, and we looked at the way that these experiments were being designed. We looked at the statistical analysis that was reported in these papers, and we looked at the type of information, how the experiments were being described. And we looked at experiments involving mice, rats, and non-human primates. And we found significant scope for improvement in each of these things. So in terms of design, very few publications reported the use of randomization or blinding. None of the publications reported the sample size justification. In terms of analysis, we found that only 70% of the publication reported, fully described the methods that had been used to analyze the data and presented the results with a measure of viability. So that means that a third of a publication are actually missing the minimum information necessary to understand the results. And then in terms of the reporting, we looked at, how the experiments were being described. We looked at the animal characteristics, and we found, for example, that, only a quarter of the publication reported both the weight and the age of the animal. Right, I think I got cut off.
Hi, you're back. You're back.
Yeah, I just suddenly got cut off. Sorry.
For a while too. I'm not sure who it was, but I can see you now.
Okay, good. All right, so there was a lot of scope for improvement, and we started thinking about what we could do to improve standards in these areas. And we decided to start with reporting, and that's how we developed the RAF guidelines, but I'll tell you a little bit more of the guidelines a bit later. Going back to the state of animal research, those findings that we found were just really not isolated. Basically, virtually every study that has looked at the quality of animal research has found the quality wanting. And these are results from the Camarade's group in Edinburgh. Malcolm MacLeod, I think you've had a talk from him recently, showing the prevalence of reporting randomization, blinding, sample size calculation in different disease area. And you can see that it's pretty low across the board. Randomization is only mentioned in about 25% of publication, blinding about 30% of publications. Sample size calculation is actually fairly consistent. It's less than 1%. So there's clearly an issue with the reporting of animal research. And it's not just the reporting, it's probably reflecting the way that studies are designed as well. Now, why this is so important, sorry, I just forgot to mention one thing. You might think that it might depend on where you publish the research. And, if you publish in higher impact factors, then that's going to be different and the research is higher quality. That's really simply not the case. These are the same data, but just presented by this side of journal impact factor. And you can see that randomization, the prevalence of randomization does not increase if you increase the impact factor, actually it's decreasing. Blinding is pretty flat, sample size calculation is pretty flat. So where you publish doesn't matter. The quality is actually pretty poor across the board. And why this is important, and that really fits into why research is not reproducible. So you probably heard of that paper. This was published 3 years ago, I think now. And that paper estimated the cost of irreproducible research and preclinical research in particular. And they actually estimated that about half of research produced is not reproducible. And then they started looking at the causes and why this was. And the causes that they identified were the materials, the study design, the data analysis and reporting, and the protocols. So in terms of what we do at the NCFURs, we really focus on the two middle one here, the study design and the data analysis and reporting. And that actually accounts for about half of the waste in animal or preclinical research. So now moving on to explain why these things are so important. And I'm going to start with randomization. So randomization is used normally to allocate animals into groups. And the method that you use is really important. So haphazard or arbitrary allocation is not random. So you have to use a validated method to randomize your animals into groups. And that could be a computer-generated list of numbers, that could be throwing a dice, flipping a coin, but it has to be something that you cannot influence. And that's crucial for two different reasons. First of all, randomization minimizes selection bias. So it minimizes the risk of systematic differences in the animal characteristics, in the characteristics of the animals that have been allocated to different groups. So if you use haphazard or arbitrary allocation, for example, you might end up with all the slowest mice in the same group, or the mice that are actually the more friendly, because those are the ones that you caught first. So that's why you need randomization, a validation method, so that you can't actually influence this. And the second reason that randomization is so important is that it actually helps you meet one of the key assumptions of the statistical analysis. That says that different groups should be drawn from the same background population using random sampling. So if you've not used randomization, you're not actually supposed to use statistical analysis on your data. And just to show you the difference between arbitrary allocation and random sampling. So I really like this graph. This was actually an internet survey that asked people to pick a random number. So they had to pick a random number between 1 and 20. And then they compared those results to a computer picking random numbers. So if you look at the white lines, the white bars are the computer. So it was a number between 1 and 20. So each number had a 5% chance of coming up. And you can see the computer, the results are actually oscillating around the 5% line. If you look at the replies from people, and we got that survey got nearly 350 responses, you can see that 17 and 7 were just picked most of the time. So 17 was picked in nearly 20% of the time. 7 was picked in a further 10 to 15%. Somehow 17 is the most random number. And clearly that's not true. 17 is not, is not random. That's just, it's all based on cultural differences. It's based on, that's a prime number. So to a human, it actually seems random, but it's just humans can't do random. That's the thing. Like we're just going to put a cultural influences on it. We're going to think about it. We're going to try to find a system. And at the end of the day, that's not random. And that's why we need to use a computer, because we're always going to be subjective. And we're humans. Humans can't do random. That's fine. We've got other skills. But that's why you really need to use a validated method on randomization and not just haphazard or arbitrary allocation. And randomization is not only important when you randomize your animals to groups. You actually have to use randomization throughout the experiment, and that helps you reduce bias throughout the experiments. There could be loads and loads of different factors that could come into play once you've already allocated your animals into groups. So, for example, you might be administering different treatments at different time of the day. So you really don't want to administer all your control animal first and then all your treated animals, because that could actually induce bias, and you might have different effect of circadian rhythm, for example. So you could use randomization at this level so that you randomize the order of treatment when you're actually administering the treatment. Same thing applies to taking measurements or performing a health check. If you're actually checking all the animals in order when you do a health check, then if you notice one side effect, you might actually start looking for that side effect in the animal that comes afterwards. So if you've done all your controls first and then all your treated, then you're going to notice more things in the treated group, and that's going to introduce bias. So again, here, when you're taking measurements or when you're actually performing checks on your animals, randomizing them before you do it, just a simple randomization so that you do it in random order, is really, really helpful. Then you've got the position of the cages on the rack, for example. So, on the rack, you're going to have cages that are closer to the door. You're going to have cages that are closer to the light. That might actually introduce bias in your experiments. We know that light has got effect on sleep levels. It's got effect of tumor growth. It's got stress has got effects on humans as well. So you need to think of that and think about, where are the animals in the cage? If you've got all your treated animals at the top, like on this picture, and all the control underneath, then actually maybe you're not measuring the effect of treatment, you're just measuring the effect of light on the animals. So you need to think about it and randomize the cages across the rack. You need to think about, if you get different staff working. So let's say that you've got a staff that's really experienced, that's training a junior staff on how to administer treatment or inject animals. And what would be really, really common is for the senior staff to do all the drug injections and all the, for the trainee to do all the controlled injections, because the controlled injections are just placebo. So they don't have saline, so it doesn't matter that much. But then if you do this, that means that the animals that are receiving the control have been handled differently. And they've been handled by someone that didn't have the same level of experience, and they might be slightly more stressed out. And again, that might introduce bias in your experiment. So if you've got several people working, sometimes you just can't do it on your own, and you need to have several people working in this, then actually just randomize who's going to get what animals and make sure that it's not, one person getting all the controls and one person getting all the treatment. So randomization really needs to be used throughout the experiment to reduce the occurrence of bias. Oops, sorry, just missed the slide. So moving on to blinding. So blinding is also used to minimize the effect of bias, and it can be used at different stages of the experiments. So you can blind during the allocation, so to make sure that you don't know what animals are going to be allocated to what treatment, and that's to minimize selection bias, so that you've got no say in what groups the animals are going to go into. You also need to use bias during the conduct of the experiments. So that's to ensure that all animals are handled in the same way, they're monitored in the same way, and they're treated in the same way. Because if you know that an animal has had a stroke, then you're actually going to look at specific side effects, and then that's fair enough. But then you're going to treat the animals that are the sham animals differently. And you might actually just, you might not look at the same side effects if you know this. So ideally, you'd want the animal care staff to be blinded to the treatment the animal have received. You also want to be blinding during the assessment of the outcome, and that's to minimize confirmation bias, because if you think you know what the treatment is going to do, you're just going to see what you're looking for, basically. So you really need to be biased at that. You need to be blinded at that stage. And then you also want to be blinded during the analysis of the data. So obviously you can't be blinded to everything. You have to know which data points are grouped together, but you don't need to know what those groups are. You could just have like group A, B, and C. Because when you're doing the analysis, then actually you might have to make subjective decisions. You might have to think about applying a data transformation if your data doesn't fit the assumption of a parametric test, for example. Or you might be handling missing data, you might be handling outliers. And if you know what that data point is, and if you know what effect your drug was going to have, then actually you're going to make slightly different decisions. So ideally, you don't want to know what those groups are when you're handling the data, and it can just be really revealed at the end of the analysis. And just to give you an example as to why randomization and blinding is so important, so this is a paper that was published in Nature in 1988. And this paper basically showed that if you dilute antiserum to human antiserum against immunoglobulin E, so that paper showed that very, very dilute solution, to the point that there was no molecule left in the solution, triggered basophil degranulation in human. And that paper was actually used to explain the memory of water theory, which is the theory that supports homeopathy. So when Nature got that paper, they actually agreed to publish it on the condition that they would send a team to the lab in Paris and observe the results being repeated. So it was a proper replication, and the Nature investigator went to Paris and they observed a replication of that study. And obviously what happened is that when the investigators insisted, oh, sorry, I'm losing my slides. What's just happened? Okay, sorry, I don't know what happened. So when the investigators insisted that the antiserum tubes were coded and randomized before being presented to the investigators, the slides for pathology were recoded and presented in a random order, and then duplicate measurements were taken by 2 different investigators, then actually there was no effect at homeopathic dilution. And that's really important, because this was not a case of fraud or anything like this. Like, the investigators really, really thought that they were onto something, and they really believed in that result. It's probably a bit too much. But, you know, they were basically seeing what they wanted to see. And when they were preventing from doing this with really rigorous methodology, when the effect just disappeared. So moving on to sample size calculation, and why it's so important to justify your sample size. And also, you know, what is the problem with small sample sizes? Because you might think that, you know, if you got a small sample size, but you're able to detect an effect, then, you know, surely you had enough, you know, enough animals to detect that effect, and you're fine, you've got a big enough sample size. Unfortunately, it's just not that simple. And I'm going to try to explain why, and I'm going to try to explain the impact that small sample sizes can have on an entire field of research. So you probably all know this, the parameters in the power calculation. You've got the effect size. That's the minimum difference you want the experiment to be able to detect. So normally that will be based on what is clinically relevant or biologically relevant. Then you've got the variability, which is normally estimated based on previous experiments. But if you don't actually have any data from previous experiments, you can actually do without the variability, and you can use the standardized effect size, for example. So there are other ways. Then you've got the power. So the power is the percentage of true positive the experiment can detect. And normally we aim for like 90 or 80 to 90% power. And that means that if you've got 80% power, if you're running an experiment 10 times and there is a true effect to be detected, you're going to detect it 8 out of 10 times. That's what the power is. And then you've got the significance threshold. So that's the probability of obtaining a false positive. And the threshold is normally set at 0.05. That's the convention. And that means that you've got a 5% chance of obtaining a false positive. So now let's think of a scenario when you've got 1,000 hypotheses that are going to be tested in 1,000 experiments. And let's say that 10% of these hypotheses are true. So in 10% of these experiments, there will be a true difference to be detected. And in 90% of the experiments, there'll be no difference to be detected. And we're going to run this experiment with 80% power. So if you start there, so the experiment where there's true difference to be detected is here in green. And if we run the experiment with 80% power, we're going to detect 80. So we're going to get 80 true positive, and we're going to get 20 false negative. We've just missed the effect. Then out of the yellow experiment, out of the 900 experiments where there is no difference to be detected, your significance level is 0.05. So 5% of those 900 experiments are going to come back false positive. They're going to come back with a statistically significant result when actually there was no difference between the group, and that's just a false positive. So in terms of what's going to be published with positive results in the literature, you've got 80 true positive experiments, and you've got 45 false positive experiments. So you end up with 125 experiments published in the literature. And that means that 36% of it is actually false positive. And that actually has nothing to do with small sample sizes yet. This is all about the null hypothesis significance testing and publication bias, because we tend to publish the positive results only, and it's actually much harder to get the negative results out. So so this is with 80% power. The issue is that basically we know that most animal experiments are not powered at 80%. And this is a very nice study which was published a few years ago by Kate Burton and colleagues that showed that in neuroscience animal model studies, the average power was actually between 20 to 30%. The median was actually 21%. And there's no reason to think that it's actually any different in any other type of research. It's just that it's been looked at in neuroscience. But the power is actually quite low in a whole range of different fields doing animal research. So let's go back to that little scenario that we had and actually run this 1,000 experiments with 20% power this time and see what happens. So out of the experiments, when there was a difference to be detected, out of those 100 experiments, we're going to detect 20. We're going to get 20 true positive and 80 false negative because we've got 20% power. Then out of the 900 experiments, when there is no difference between the group, there is no difference to be detected, a significance threshold does not change. It's still 0.05. So 5% of those are false positives. Sorry. So now, in terms of what we're going to see in the published literature, we've got 20 true positive and 45 false positive. So out of 65 experiments that are going to be published in the literature with positive results, 69% of it is false positive. And that's really, really scary when you think of it that way, because that makes you think that you just can't trust anything that's published in literature. But that's what happened when you've got small sample sizes in an entire field of research. Okay, so just to recap on the power calculation, I mentioned the different variables that are used for the power calculation. So you've got the power, the variability, the significance level. You also have the signness of the test. And then if you've got 5 of these 6 parameters, you can calculate the other one. So normally the way that you should be using a power calculation is by using your signness, your power, your effect size, your variability, and your significance level, and then calculating the number of animals per group that you need. But obviously you could actually do it the other way around. You could decide that you want 6 animals per groups, and then you can say that this is the effect size that you wanted to detect all along. And you know, we're a grant funder, so we know that people do this. And in the UK, what we do now, as part of the grant application, we actually request a justification for sample size. We want to see the power calculation, but crucially, we want a justification for the effect size. And that's really, really important because that just sets everything. The power calculation on its own is completely meaningless if you've not set an effect size that is, you know, biologically relevant or clinically relevant. And the way that you could go about it could be, for example, you know, if you're working with a stroke model, so you could consider that, you know, the drugs that are actually currently in the clinic reduce the symptoms by about 30%. So you're setting your effect size at 30% because you're not interested in a new drug that's going to have less effect than what's currently in the clinic. So you could decide that, okay, 30% would be clinically relevant. This is what I'm going to go for. This is what I'm going to look for. If you're doing much more basic research, for example, you're planning to silence a gene using RNAi, so you're going to have to decide what efficacy will be satisfactory. So do you need to knock down, you know, 50% of the gene? Is that going to be enough? Or do you need to knock down 90% of the gene to actually see an efficacy? And you're going to have background information about the gene, about its function, about its abundancy, the turnover time of the gene product or other gene product that could have overlapping functions. So you're the scientist, you know everything about that gene, and you should actually use this information to decide what would be biologically relevant. No statistician is going to be able to tell you that. It's really for you as a scientist, you know, knowing that science to decide what would make sense. And really, so the effect size is the minimum difference that would convince you that that work is worth taking forward into further research. The significance level is meaningless. It's just going to tell you whether you've achieved that effect size by chance or not. But, you know, you actually need to set the effect size to start with. Okay. Whoops. Sorry, I'm really struggling with my slides for some reason. Another thing that impacts on the sample size is welfare. So this is a study which looked at infection and catheter blockades in rats that had been fitted with catheters long term. And you can see that, you know, they're comparing those that had a sterile surgery to those that did have a non-sterile surgery. And you can see that a lot more rats in the non-sterile surgery group had infections, blockade of the catheter. Survival was actually 3 out of 6 rats for the non-surgery group, the non-sterile group, sorry. Whether for the rats that underwent the sterile surgery, none of them got an infection or a catheter blockade, and 6 out of 6 rats survived. So basically, when you think about your sample size, first to start with, your sample size is the numbers that you need in your analysis. So if you're expecting attrition or mortality or anything, you're going to have to factor that in. So if you're expecting half of your rat to die, that means that you need to double up your sample size to start with. Another thing that impacts on the sample size is the viability. So if your rats have an infection, and there's a common myth that rats don't need sterile surgery because they don't get infection. No, they don't get visible signs of infection. I think that's very, very different, because they will have an underlying infection. They will have mediators that are upregulated, and that might have an effect on, first of all, what you're measuring, but also that's probably going to add viability, which means that if your viability is bigger, you're going to need a bigger sample size to detect the same effect. So welfare actually impacts directly on the sample size, because you need bigger sample sizes. You need a lot more animals to do your experiments if you've got poor welfare. That's another example of welfare impacting on the sample size. So this time, this is a comparison between two groups of dogs. This was just measuring sodium excretion in these groups of dogs. And the control group, the group on the left, was just a normal control group. The treated group had training and enrichment. And you can see that the viability is just completely different in these two groups. So the control group that had nothing has got a lot of viability. The group that was trained and had enrichment has got a lot less viability. And that's another thing. I mean, presumably those dogs were a lot less stressed out. And that has impact on the viability of the urinary sodium expression. So that's not something that you might think about, but basically welfare, stress level are going to have an impact on your experiment. So if that's not directly biasing the results, it might still induce or cause a lot more underlying viability, and then that's going to push up your sample size. All right, so I've got to the end of part 1. So if you want to ask any question. I don't know how that's going to work. Olavo, are you going to read questions?
Yeah, you can ask questions. I mean, you can either raise your hand and we'll give you voice and microphone. Otherwise, you can send questions via a chat to just copy us all when you're doing this. We have around like 20 people watching here, some people watching on Facebook. You can ask on Facebook. We'll try to look that up. That's Zoom. If I can start with a couple of questions. Just the other day I was reading, I think it's Fasting and Altman. It's a paper on how to design industry experiments. I think it's 2002, and it's very, we're in 2019 now. It's like 17 years later. We're pretty much talking about the same thing. I'm not sure a lot has changed if you look at like Malcolm's paper, etc. Like it's not like people are just like starting to randomize like crazy. It's improving a bit. Like why has uptake been so slow? We've switched the generation already. Like Doug has passed away, and we're still insisting on it. Why do you think it's hard? Because some of those measures are easy. We can talk about the hard ones later, right? But like randomizing is easy. What keeps people from taking this on?
I think, first of all, that, Altman and Fasting paper was written in 2002. And I think at that time, Doug Altman is a clinician to start with. And I think those were measures that, were commonly used in clinical trials. But the animal research field was actually far behind. And there was no awareness that these things were actually important, that there was a problem with animal research. When we came out with that survey that I presented, so that survey was published 10 years ago, and this was news to everyone. This was the first survey that actually looked at the quality of animal research, and everyone was actually really, really surprised by the results. And we weren't expecting anything that bad either. So I think that the problems were actually just not known. At the time that we published the survey in 2009, no one was talking about reproducibility. The first papers that actually started talking about reproducibility probably came out in like 2011, 2012. So I think it's just been a slow realization, first of all, that there is a problem. And then, once you actually accept or realize there's a problem, then you need to think about, okay, what are we going to do to improve it? What are the low-hanging fruit and so on? So it's been a slow process, but I think things are really changing. When I present that data now, no one's surprised, whether 10 years ago, people really, really were surprised by this. So it's definitely been improving. It's just slow. And the animal research field is behind the clinical research field. I don't know exactly what the reasons are. There's lots of factors in play. You've got research culture, you've got the pressure to publish and so on. But yeah, the animal research is behind clinical research for that.
Okay, so we're waiting for somebody who's going to ask a question, but Sileni, are you writing it? Are you okay? Yeah. So this is what is your area of research? You're referring to behavioral experiments in rodents. So her question is like, we've found very big effect sizes in terms of standardized effect sizes in some areas of research. I think, as far as I know, she works with like forced swimming in animals. I could be wrong. But antidepressants and animal behavior. So like you have like hedges G of like 1 or 2 or even 3. Could that explain the low sample size? Or maybe I actually like, if I can put the question the other way around, I mean, are those effect sizes very big because power is low? And because, are you referring to the literature or to your own experiments? Because I mean, you could see this the other way around. Like if your power is very low, you only publish when you have like very, very, very big effect sizes. So I mean, is this the effect size driving the low power, the low sample sizes, or is it actually the other way around? Those sample sizes are driving the high effect sizes. Yeah, so she's referring to a meta-analysis, so I guess I'm
actually not sure what the answer is here. I think, I mean, with the low sample size, you could always get, you know, a result by chance and then detect a statistical difference for a very small effect size just by complete chance. So I'm not sure that you've got a direct correlation between the two. You would think that with a very small sample size, you can only detect the big effect sizes, but then you've got all the other bias that go into this. And if you've got an experiment that's not randomized and not blinded, well, actually, you're going to see those big effect sizes. So I think you can't just attribute it to the small sample size only. I think there's loads of different factors at play here. And at the minute, because there's so many different factors, we've not really identified which ones was the main contributor in a sense.
Okay, I have a second question, but maybe it fits better with the second half, so I might hold on for that one. Anybody else at the moment? Facebook? Anybody checking? No. I could make the second question now, but well, actually, like making a setup for that. I mean, you mentioned that a lot of this came from the clinical field. Are we missing anything that's different? Because I mean, sometimes I do wonder about randomization, for example. I mean, we do speak a lot of that. I'm not arguing against it because I mean, it's very difficult to implement actually. But you could argue that, I mean, odds of like taking slow animals, whatever, are okay. It could happen. It does violate your assumptions, but it's probably less drastic. I mean, it has less an effect. Like in the clinics, you can see like if a doctor is choosing that, okay, this guy should be operated on, this doesn't, this should get medical treatment. I mean, this completely messes up the whole thing. I mean, it could be, I have an intuition that it could be less important for animal research. I'm not sure. I don't think we have the data for that. But I mean, are we overrating something or are we underrating something? I mean, is that something? I mean, we do import a lot from the clinical literature. Can we be missing something vital here as well? And I don't know.
I think so. I agree with you that there's no or not much empirical evidence of the actual impact of randomization in animal research. It's just not been tested that clearly. But what we know is that it's a clear risk. And you get that example of like if a clinician is just picking the patient that is going to operate on that. But after I used to do animal research and I used to work on antiemetics. And, I was working with ferrets, and with ferrets, you don't, you basically get what you get. So sometimes they're quite small, sometimes they're much bigger. The weight was definitely not harmonized. And, when you get a small skinny one, you just don't want to give them the chemo. You're like, oh my God, like that skinny one is just not going to do well with the chemo. So you always have your instincts that, this one, you just don't want to give it to this one. That this one doesn't look well. You want to give it the drug, or you want to give it the control. And so it's much easier to have a blanket coverage saying like, right, I'm just going to protect myself against all those things. And if I'm doing it and actually wasn't needed, well, it doesn't matter that much because it's actually really easy to do a randomization. So I'd say that it's much better to be on the safe side and use it. Then in terms of whether we're missing something because we're just doing what the clinical field is doing, I don't think this is what we're doing. And I think that's probably why we're actually behind, because we can't just apply what the clinical field has done. And in terms of statistical analysis, in terms of addressing bias in experiments, I think animal research has worked out, its own way of addressing bias, for example. So, in a clinical trial, if you want power, you're going to have to push up your sample size. There's nothing else you can do. Like, you're working with a population that's heterogeneous by definition. You're going to have to push up your sample size. In an animal experiment, if you're going to push up your power, there are actually other things that you can do. It's much easier to reduce variability because you can control this. And you can control the characteristics of the animal that you're getting. You can also control for variation. For example, if you're doing your experiments on several different days, you could just easily use a blocking factor, and that's going to take some of the variability into account. So there are different strategies that you can use in animal research. And we're not just applying the findings of the clinical research field blindly. I mean, all those examples that I gave about randomizing throughout the experiments, these are things that would not necessarily apply to a clinical trial. You don't have to worry about the position of the cage on the rack for a clinical trial, for example. So we are working on, we're working out kind of like what are the factors in animal research that you need to be careful about and you need to address.
Cool. Well, do we have any more questions? Not now. So yeah, I guess we can move on. And I have one, I think, a very important question concerning the project itself, which might be, but I'll leave it for later.
Okay. All right.
Just a minute, Olavo. We have a question. Please, okay. Just a minute.
Yes, so but this is that's actually a good question in terms of so she's asking about how repeating experiments versus repeating samples versus changing the sample size within an experiment. I guess that's what she means. So like. And that is actually, to me, an important question for people in basic research. I mean, whether you should do like a lot of different experiments with like some animals versus like one experiment with like a lot of animals, and how do you see that? How do you analyze that? And
right. Okay, so what's the best strategy?
Yeah, I mean, instead of like increasing the sample size within a single experiment, actually doing independent experiments multiple times.
Right. So, well, I think, the people have different views on this. My view is that, first of all, you want an experiment that's appropriately powered. So, if you're doing one experiment, you actually power it so that you can detect the effect that you think is of interest. And then, if you're actually trying to get more validity, you want to make sure that your results are really robust. I wouldn't necessarily repeat that same experiment in the same lab over and over again, because I think that's probably a way. What I would probably do is try to, increase the variability a little bit. So repeat it in a different species or in a different strain, or, just get the protocol to a collaborator that can actually repeat that experiment in another lab. And there's been quite a lot of research coming from Hanno Werbel in Switzerland, showing that, if you actually repeat the experiment or do multi-center experiments, when you've got experiment that's done across 2 or 3 different labs, then the results actually are a lot more robust, and you can, because that you can actually take into account all the different variability that you've not thought about, because you've got if you're actually working in different labs, you've got different factors that are going to be different. You're going to have different level of humidity, different staff working there, so all those different things that might induce variability and might actually bias the results are going to be different in those different labs. So once you get results, once you get a positive signal, you can be fairly confident that this is the effect of your treatment and not anything else.
Well, happy to hear that. We're doing everything in 3 labs, so I guess we agree on that one. So yeah, I think if nobody else is around, nope, I guess we can move on and we'll leave the rest of the discussion to the end.
All right. Okay. Okay, so a lot of what we do at the NCFRs is develop resources to help researchers. So we've got a whole battery of resources on experimental design specifically. So I've put the URL here at the top of the slides. All those resources are available freely to anyone who accesses the website. And the 2 resources that I really want to talk about in more detail today are the Experimental Design Assistance, or EDA, and the RAF guidelines. So starting with the EDA, So the EDA is an online software which was developed to guide researchers through the design of animal experiments. So basically, you design your experiment in the software, and the software provides feedback, and it's like having your own statistical assistant with you when you design the experiment. The reason that we've done this is because we realized that there was definitely a shortage of statisticians that could advise on the design of animal research. Very few researchers have a statistician that they can talk to in their own institution. So we wanted to provide a system that could actually support researchers and something that was scalable, because we just can't train enough people to do this. So the EDA provides advice that improves the experimental design. It provides recommendation for the statistical analysis. And there's also dedicated support for power calculation, randomization, and blinding. And so this is what it looks like. So within the EDA, every experiment is represented as a diagram. So I'm just going to talk you through that diagram. It's just a very, very simple comparison to give you an example. So it's a two-group comparison. In this experiment, you've got animals that are allocated to two different groups, group 1 and 2. Group 1 gets a vehicle injection, group 2 gets a drug injection. Then a measurement is taken. In this case, plasma glucose levels are measured. And then the data is analyzed. There's only one variable. The independent variable of interest is the drug with two levels, vehicle and drug. So that's a very, very simple two-group comparison, just to show you what that looks like. The diagrams include three parts. So you've got the practical steps that you're going to do in the lab. So you've got animals and you split them into groups and you're giving them injections and you're measuring them. And then you've got the high-level information about the experiment. So that's the gray and black nodes. And so in there you've got information about the animal characteristics, you've got the experimental units, and then the pink or red in nodes about the analysis. So you've got all the detail about the analysis and the variables that go into the analysis. So your entire experimental plan is actually represented in one diagram. And those diagrams are a lot more explicit than text description. Because if you think of blinding, for example, in a text description, you're never going to have that information. If you're lucky, you just get experiments done blind, but you don't actually know who was blinded to what part of the experiment was done blind, what part of the treatment, is that the treatment that was blind, that you were blinded to? Was that the, at the level of the measurement? You just don't know this. So the diagrams are because you can indicate that information on the diagram at the level of randomization, the measurement, or the analysis. So you can be really explicit as to what's going to be done at each stage. And that's the information that's inside each of these colorful blobs. So we call them nodes. So this is the properties of the allocation node. So in there you can indicate your randomization strategy. In this case, that's a complete randomization, or your randomization procedure. So how you're actually going to do the randomization. Are you going to flip a coin? Are you going to use a computer-generated list of number, or you're going to use the spreadsheet that's generated by the EDA. So you can indicate that. So you got a level of transparency, and it's a lot more explicit that you normally have access to. And then the diagrams are actually machine readable. So the computer with the software uses computer-based logical reasoning to provide feedback. So the feedback is always tailored to the experiment that you're trying to do. So for example, this is a prompt telling you that you've not included any nuisance variable on your diagram. So on this prompt, you can read about nuisance variable, what they are. You can read about common examples of nuisance variable in animal experiments. So things that you have to consider, for example, the cages of the room, or if the animal had no house together. So you've got a lot of information to just prompt you to think about what you have in your experiments and what could induce variability and what you should actually include so that you reduce that variability, so what you include in design. That's another example of a prompt providing feedback, telling you that you need to indicate the blinding status during the assessment of the outcome. So on this prompt, you're going to find information as to why blinding is important, the different stages of the experiments that need to be done blind, and the different ways that you can blind different stages of the experiment. So it's just giving you all this information so that you can make an informed decision, basically. And so at the end of the process, when you've designed your experiment in EDA, you've got your feedback, you've got your recommendation for analysis, You can actually generate a report. So that's just a simple PDF report. In the UK, we actually use this as part of the grant application or as part of the ethical approval for the experiment. So that report summarizes key information about the design. So it summarizes the objective and hypothesis, the animal numbers, and the justification for the sample size, the steps that are going to be taken to minimize the effect of bias, like randomization or blinding, the primary outcome and the secondary outcome measures, and the planned statistical analysis. And then at the end of the report, you get a diagram as well. So you got a visual representation of what you're going to do with that experiment. So those reports are standardized. Again, they're tailored to any experiment. So if you've got 25 groups, then you're going to have your 25 groups in the report. If you've got different measurement steps, then you're going to have the blinding status for the different measurement steps. So it's all tailored, but it's all standardized as well. So as a referee or as a panel member, when you see that report, then you can quickly go through it, and you get a really good idea of what design is going to be done and what steps are going to be done to ensure rigor and quality. It's also very useful as a researcher when you do it, because you can just generate the report, and if you've forgotten any information, then you've got a big line saying information not provided. So it's very easy for you to actually spot what information you still need to provide. You can go back to your diagram, get feedback from the system, and provide that information. So that would be a workflow when you're using the system. So you start by drawing a diagram, you add details into the node properties, you critique your diagram, you get feedback from the system, and you probably will go through the steps several times until you're actually happy with your design. The system is not going to force you to do anything. It's basically telling you, what that thing is wrong and how you can improve it. And then you can decide. You can decide what you do with it. So once you're happy with it, you can choose a method of analysis, and the system will actually provide suggestions for this. And then you can, once you've decided on the method of analysis, how you're going to analyze the data, you can calculate your sample size. And there are a couple of sample size calculators in the system with a lot of guidance on how to use them. Because there are loads of power calculators online. You can use C-Power, you can use power calculators everywhere. The issue is that if you don't know what you're doing, actually deciding on the parameters that you're going to put in the power calculation is not that simple. And actually, to start with, deciding on what calculator to use is not that simple. So we've included a lot of guidance in the system. We only offer 2 power calculators, but with guidance on the parameters that go into this. And we also have a decision tree that actually helps you decide what calculator you need to use for your power calculation. Then once you know how many animals you're going to have per group, you can get the EDA to generate the randomization sequence. So you actually generate an Excel spreadsheet that's got your animals and what group they've been allocated to. And it's not going to give you that spreadsheet. It's actually going to send it to the person that's going to help you with the blinding. So, as the person designing the experiment, you can remain blinded. You can remain unaware of what animals is going to be allocated to each group, because you don't even see that spreadsheet. So then the person receiving that spreadsheet can just enter the unique animal identifier and then keep that safe. And then they can help you with the blinding. They might be injecting the animals themselves, or they might be preparing the syringes for you when you inject the animals. But they're going to be holding the randomization sequence and keeping it safe so that you can remain blinded for your experiment. You can share your diagram, so you can actually work with the collaborator on the diagram. You can actually do that instead of the system of the process. So you can just share your diagram so that your collaborators can see exactly what you're doing, and they've got all the details about each of these different steps. As I mentioned, you can export a diagram report, which is that simple PDF document. And then, hopefully your experiments get funded or approved, and you can actually do the experiments. And then there's a few fields that you can go back to and update afterwards, after you've done the experiment. So those would be things like, you've had some attrition, and the number of animals that you wanted to use is actually different from the number that you ended up analyzing. So you can actually update that field, and you can provide a reason for why these numbers are different. And that's really, really useful, because you're probably going to be writing up that experiment 3 years down the line. So having all the detail in one place with all the, the different reasons for changes is really, really, really useful. So the EDA has got several benefits. And so first of all, we developed it to improve the reliability of published results. This was actually the primary goal when we developed that system. And the EDA really does that by addressing obvious sources of bias. It's going to help you identify sources of bias in your experiment, and it's going to give you advice on how to address it and how to incorporate this into the design of the experiment so that you can avoid that bias or reduce it. It also promotes better understanding of experimental design, and it raises awareness about why these issues are important. So the EDA is not a black box telling you how to design the experiment. All it does is just every time there's a decision point, it's going to provide you feedback, and it's going to give you the implications of going one way or the other, basically. So you just get the implications of these decisions, and then based on the implications, you can actually take an informed decision. But every time you get feedback, you learn something new. One of the key things with the EDA is that, when we developed it, we knew that there were books about experiment design, and that information was available in lots of different places, but animal researchers were just not using them. They were not reading these books because they just did not have time. As an animal researcher, you just don't have time to step away from your research and learn about something new. You have to produce, you have to write grants, you have to publish. So that system was designed so that you can actually learn about experiment design while you're designing your own experiment. So you basically learn as you go, but you don't need to take time away from your research to do this. The system also facilitates assessment of the experimental plan with an explicit description. So as I said, those diagrams are a lot more explicit than text description. I guess that your protocol that you're going to be using for your application studies are pretty descriptive as well. But if you compare this to like a normal paper, then an EDA diagram contains a lot more information. And that transparency is really, really useful because it's useful if you're going to be using this as part of a grant application. That transparency is useful for the peer reviewers, for the panel members, for the ethical review board who's going to look at approving your research. And you can even, submit a diagram or submit a report with your manuscript when you publish it. So that transparency is going to be useful to the readers of the manuscript as well. I was having a chat with the lab earlier saying that in some cases for your application studies, it was hard to identify which one was the control group, when that would not happen if you've got an EDA report or an EDA diagram. It's going to be very, very obvious which one is the control group because you have to identify which one is the control group. So it's got a tick and you know which one it is. The reports are also useful as a form of pre-registration. So you can use it as a form of like soft pre-registration, if you will, if you actually do it yourself and you keep it safe. But, at least you've got something registered somewhere, even if you've not shared it publicly. But if you want to, pre-register it properly, you can actually just put your EDA diagram, you can just download the file and put that on Figshare or on the OSF. And then that's, time-stamped, and you've got your entire plan pre-registered. And that's just really easy because you've designed the experiment already, you've got all the information in the system. All you need to do is just download the file, just stick it on the Figshare, and that's it. I think what the system does is that it promotes a more careful consideration of the experimental plan. So, we actually want people to spend more time planning their experiments. Once you've collected the data, it's just too late. You can't change anything. So we really want people to spend that time planning. And a question that I often get is, okay, how long does it take to plan an experiment in the EDA? To be honest, if you really know exactly how that experiment is going to be done and you've got all the, everything identified, it really doesn't take time. It takes me 10 minutes to build a diagram. That's not what takes time. What takes time is building a diagram, getting feedback from the system, thinking about that feedback, discussing that feedback with your team members, your collaborators. And that's going to take time. Yes, you're probably going to spend a bit longer designing that experiment. But at the end of the day, that's going to be an experiment that's really designed robustly. And you're going to have all the feedback that you need before you collect your data. And that's going to be much, much better quality data. So that's time really well spent. And the diagrams really facilitate discussion. We know that the diagrams are actually used in lab meetings. And normally what's used in lab meeting is your results, because you've got something to show on the screen. You can show your graphs and it's brilliant. And then, it's very visual and you can discuss the results. That's what you share in lab meetings. Well, now you've got a diagram that you can share that represents your experimental plan. So if you put that on the screen, you can get your lab members to comment on it. And then you can see that people start asking much harder questions. Rather than just if you describe your experiment orally, people are still trying to understand how many groups you have. There's no way that they can actually give you proper feedback. But if you put a visual representation on the screen with everything identified, remember that the variables are identified as well as visual representation, then people can start asking the hard questions. They can ask, what about this data transformation? How are you going to, transform that data? Why are you doing this transformation? What about this variable? How come it's not included in the analysis? And so on. So, you can actually get constructive feedback from your team. And your lab members know the science just as well as you do. So it's actually really, really useful that you can create an experiment together. And that's, the EDA diagrams and the visualization is actually a tool that enables that discussion and enables that collaboration. And so just to give you a bit of information as to how that system is being used. So we've got, we released it 3 years ago, and we've got 7,000 accounts in the system. So 7,000 users using it. We get about 3,000 website visits a month on the website. It's currently recommended by, I actually need to update that slide. I've just got the new numbers. It was 80 different organizations in 17 different countries. And it's mainly recommended by funders because funders can see the benefit of it. So all the funders in the UK, like the Wellcome Trust, the MRC, the Academy of Medical Sciences, recommend the EDA because they can see that it's going to lead to higher quality research. And funders have got a vested interest actually paying for the research. So they want to see something, they want to see quality data at the other end. In the US, the NIH and the NSF actually recommends that applicants use the EDA when they prepare the grant application and when they design their experiments. And so this really shows that, the funders actually behind it, they're serious about getting better quality data. They're serious about getting reliable results. And this is a system that you can use to help you do that. Okay, so moving on to the ARRIVE guidelines. So the ARRIVE guidelines are about reporting animal research. So, once you've designed your experiment, you've got your results, you're writing up your research, that's where the ARRIVE guidelines come into play. So those were developed in 2010, so 9 years ago. And it's basically a checklist that contains the key information that's necessary to describe a study in a comprehensive and transparent manner. So the original guidelines had 20 items that cover things like internal validity, reproducibility, like method reproducibility, and also the context and the relevance of the study. These guidelines were developed as a consensus between scientists, statisticians, and journal editors, as well as research funders. And as I said, they were published in 2010. I've talked about the internal validity and the importance of it already, so I just wanted to give you a couple of examples on items that relate to method reproducibility and why they're important. So just to give you an example, we've got an item of the guidelines on housing. And that may seem like a lot of information to report. I mean, who reports the cage type of the housing or the bedding or that, that kind of information. But it's actually really, really important. And that's an example of a neuropathic pain model when the actual model development is based on the bedding texture. So depending on the bedding, the texture of the bedding, then you can see the symptoms or not, and you can actually develop a user model or not. So that's actually quite a key detail, but that's something that no one was thinking about. And I think this was probably discovered by accident. And that's why it's so important to report that information, even if you think that is not that relevant. At the end of the day, that's the housing of the animals. The experiment is about the animal. Every factor or every parameter of the housing is probably going to have an impact, including things that you'd never think about or you don't even know. So, the things like the animal facility, the details about the number of cage companion, the cage tongue, any that type of detail, it's easy to report. You all have this information. You might as well include it. If you can't include it in the body of the manuscript, then you can include it with the data, or you can include it supplementary information. But that information is going to be really, really useful for readers of the paper. And I think, especially with the type of experiment that you're doing, those replications, that information was probably not included in the original papers, but then it's really, really important for you to include it because that will explain, different results. And if you've got different results in the 3 different labs doing the replication, then actually you might be able to find the explanation with that information. So it's really, really important to include it. Another example at the bottom of the slide is about husbandry. So in terms of husbandry, we recommend that you report the breeding program, the light dark cycle, the humidity, the temperature of the room, so on. And again, that's a lot of information, but that has an impact. And we now know that low temperature actually induces suppression of the anti-tumor response, and that promotes tumor growth. So if you're doing a cancer study, the temperature of the room is actually really, really important. And you know, in most papers, that's not reported. And that might actually be a factor that explains differences between different labs. In terms of endorsements, the RAV guidelines are actually endorsed by thousands of organizations across the world. So about 1,000 journals, but also funders, universities, learned societies. So in terms of journals, all the main biomedical research journals endorse the guideline, including all the big ones like Nature, Science, Neuron, and so on. Most of the funders in the UK also endorse the RAV guidelines and make reporting in compliance with the right condition of funding, and we've got quite a few funders outside the UK that are still doing that as well. Learned societies have been really, really helpful in kind of disseminating the guidelines as well, and that includes societies in Brazil. So FASB, for example, endorses ARRIVE and actually try to promote their use. CBCAL as well endorses ARRIVE. CONSIA endorsed ARRIVE in 2014, I think, so it's been going on for a while, and try to push it through like IACUC committees and so on. So those guidelines are really, really supported by all the different stakeholders of preclinical research. One thing that I wanted to mention as well is that we've got a lot of translations, including Brazilian Portuguese. So if you want the Brazilian Portuguese version, you can just go to the RAV website and then you got a link to the translation. So that might be useful. And just like one last point about the RAV guidelines. So I said that this is about reporting. But really, if you're going to have to report something, I highly recommend that you actually have a look at it before you do the experiments. Because first of all, you've got, in the RAV guidelines, you've got the items about internal validity. So that's something that, you need to think about before. It's fine. You can still report that you've not done it, but ideally you want to report that you've blinded and you've randomized properly and you've used an appropriate method of randomization. So checking out the guidelines before you start your experiment is actually really important, because you can make sure that you're going to be doing it in a way that you're going to be happy to report, basically. And I had a chat recently with someone that was involved in the cancer reproducibility project, and he actually told me that he should have used the ARRIVE guidelines for the pre-registration of the study, because that actually would have really, really helped making sure that everything was going to be there from the start. So that's something that you can think about as well. Like, the RAV guidelines are about the manuscripts, but actually using them from the start really make your life easier down the line. And I just wanted to show you this study, which was published a couple of years ago from researchers in Switzerland. So this was a survey of in vivo researchers in Switzerland. And those researchers were asked about, how they conduct their experiments and what they report in the publication. And they were asked questions about 7 different measures that they use in the conduct of the experiment. So allocation, concealment, blinding, randomization, sample size calculation, inclusion, exclusion criteria, outcome measures, and the analysis plan. And they were asked whether they actually used these measures during the conduct of the experiment and whether they report these measures in the manuscript when they publish their results. They were also asked whether they knew about the RAV guidelines or not. And what the researchers found is that, about 50% of the participants knew about the RAV guidelines. And actually, when they looked at the internal validity score, it was higher in people that knew about the RAV guidelines. So people that knew about the guidelines actually tended to use one extra measure compared to people that didn't know about the guidelines. So that, again, it's only a correlation, but that highlights that actually it will have an impact on how you design your experiment as well. And it's a very useful resource to pick up from the start rather than wait until you actually write up the results. And also, we've actually confirmed this result in a sample of international researchers. So we've not published this yet, but we've actually run a study at the Iniciativa using random sample of researchers from PubMed. And we've seen the same correlation. People who know about the RAV guidelines actually design better studies or more rigorous studies. And we have updated the guidelines. So this is actually still work in process. We've been working on this for 3 years now. And we've recently released the draft updated ARRIVE guidelines, so ARRIVE 2019, on bioRxiv. And there's actually 2 different documents. One is the actual ARRIVE guidelines with the updated checklist. And we've also released an explanation and elaboration document. So the main departure from the original guidelines is that the items have been reorganized. So it's more obvious, the crucial information is highlighted. And it's also prioritized in 2 sets. So we've actually prioritized the most important item. We've prioritized the items without which a reader or reviewer can't even assess whether the results are going to be reliable or not. So it's all about the reliability of the results. So we've highlighted the essential 10, which includes study design, sample size, inclusion-exclusion criteria, randomization, blinding, outcome measures, statistical methods, experimental animals, experimental procedures, and results. So these are really the key item to start in priority. And we know that journals don't necessarily have the resources to check for all the 20 items or 21 items now of the RAF guidelines, but that's why we've actually prioritized the items in this way, so that it can actually be an easier way to start, basically. So everyone can actually work with the first 10 items in priority. And then once these are currently or commonly reported in all papers, then you can start thinking about implemented the other items. We've also included a couple of new items as well compared to the original version. So we've got protocol registration and data access. These were things that were not necessarily considered 10 years ago, but it's now recognized that it really adds value to your study. So they're now included in the RAF guidelines. And we've, as I said, we've published an elaboration and explanation document. So that document is a very, very useful resource. In that, it basically go through each of the item one by one and explain why the item is important, how you can do it. You get an explanation for a lot of statistical concepts, and it also provides examples from the published literature on how this information can be included in manuscript. So it's a really, really useful resource. And as I said, you really want to be reading this before you actually do the experiment. And that's it for me.
Great. So we'll have people praising the DA in the comments and saying that, yeah, they think it's awesome and they've been using it in courses on animal research, etc. We're open for questions again. If you want to, again, you can either raise your hand and Juliana will put you online, or you can write by text. If I can start the questions again as people are writing. I do think EDA is a very cool tool as well. That said, I think that people who are very used to this think it's a very cool tool, but there's a lot to learn there. I mean, it's not easy to start from scratch. And of course, there's like a lot of community. You have to know about the concept, right? You have to know about like random allocation, sample size. You have to understand variables. You have to. So I mean, there's a lot in it that you go over before it actually becomes a useful, easy tool. And again, I do a lot of teaching as well, and it has to be a step up, right? I mean, people in the reproducibility literature have been talking about, I mean, you can't change all at once. I mean, you have to go like small step by small step. So in that sense, what's your step up recommendation? I think you're trying to do something similar to that with the Arrive guidelines, but even the Arrive 10 is like, 10 is a lot. Like if you have to go 1, 2, 3, 4, 5, what's your suggested plan of like improving things one at a time?
So I think with the RAF guidelines, yeah, 10 is a lot, but those are actually really, really crucial. So we've prioritized it. We actually wanted the minimum number of necessary items, and we got to 10, and you can't reduce that list. So 10 is a lot, but they're all interlinked as well. And as I said, a very useful resource that we've provided is the explanation and elaboration document, because that actually explains what these things are. So it's really, really useful for, even a junior researcher who's just like starting, not knowing anything about it. Actually, that, really should be compulsory reading. I actually forgot to mention it on my slides. So we're currently road testing these 2 documents. So as I said, they've been published in draft, but that's not the final version yet. So we're doing road testing. We start in September, and that's going to go until the end of the month. So if anyone is currently writing a manuscript, and they're willing to just use the new Arrive guidelines and the INI as they're writing it, then they can contact me, get in touch with me, and see whether they can be included in the road testing. What you get from it is that, we'll actually review your manuscript, so we'll be able to give you feedback that can actually help you improve your manuscript, and you also get an Amazon voucher. So yeah, please let me know if you've got a paper that's basically on something that's literally ready to go, because you're going to have to share it with us before the end of the month. Just one thing about the idea, you mentioned that you need to know statistical concepts before you start. I would disagree slightly. So we've actually done a lot of work on the EDA recently to improve the usability of it and make it more user-friendly. And what you can do with the EDA is just like, start your diagram by putting the practical steps in the lab. So you don't need to do anything. You don't need to talk about a parabol. Just put, you've got a group that's going to be split into 2 or split into 3, or how many different groups you want to have. Maybe it's not even going to be split. And then, that group is going to receive an injection and then a measurement. And stop there, critique it, get feedback from the system, and the system will actually help you build the rest. So, if you don't, the idea is really, don't try to guess anything. If you don't know, don't do it. Just get feedback from the system, and the system will actually help you. Same if you've got a field that's blank, you just don't know how to answer, you don't know, when you're actually looking at the properties, if you just don't know what the options are, you don't even answer the question, just leave it blank. The system will actually help you with that.
Good. Anyone else there? Not now. Juliana, is there some?
No questions here. Olavo.
I'll move on to my hard one. So, again, this directly has to concerns our project. So we're trying to do this 3 independent replication thing, which are supposed to be as independent as possible. I mean, we're trying to do this naturalistic estimate of how what's your best estimate of 3 labs independently trying to reproduce this. And there's actually a fuzzy line between, you could argue that, okay, maybe this 3, there's a fuzzy line between what's minimum and what's recommended and what's just optional, and each lab would do it their own way. So, you could argue that maybe blinding is minimal, or minimization is minimal. Animal, I don't know, environmental enrichment might be recommended, but maybe not as minimal. And some stuff how much bedding you put on the cage, or whatever little variable. There's no right answer if it's 2 or 3 or 1 centimeter of sawdust. Well, maybe there is it, but I don't know. I'm sorry. But so, and again, we want to do this as independently as possible without compromising validity. So, we wouldn't want to let a lab not do it blinded, for example. What's your minimum set? What we discuss this all the time. It's kind of philosophical, but what do you enforce as this is minimum standard practice versus what? Okay, I would do it that way, but I assume that it's okay if you do it somewhere else. Do you have any clear line? What are your minimum requirements you wouldn't let a lab do without?
So are you talking about design, or are you talking about welfare?
I'm talking about, welfare. I think you have a better justification in terms of, okay, the law. Brazilian law requires this. Sometimes, you have to exercise your animal because, you can't do it. For welfare, I guess we can let it. Each experiment gets evaluated by its own institution's ethics review board. So, actually that we have to third party. It's an interesting question, but I don't think it's up to us. But in terms of design, in terms of getting a robust result, okay, this should be blinded. Sometimes it's up to us. And we want to be as minimally invasive as possible because we want to keep the independence. If you want to say you have to do it this and this and this way, something gets lost because it's not but there is a minimum set, and the extent of the minimum set is a topic of course of discussion. So what do you think is okay, this and this and this should be enforced, and no lab should do without the measures? I don't know.
I think about that. Yeah, definitely put randomization and blinding in that minimal set. I'd probably put the inclusion criteria as well. But then I think the rest would probably depend on the actual experiment. And this is something, I used to be part of a consortium called Multipart, which looked at doing a multi-center study in stroke research. And we've had endless discussion about, what should be in the SOP and what do you leave out of the SOP. And I think we tried to harmonize as much as we could. But at the end of the day, we had the same thing. The experiments were being approved in different centers. So, you might want to say, ideally you'd want all the animals to use the same, all the experiments to use the same anesthesia, for example. But actually different centers had different preferences and were recommended different type of analgesia. So we ended up deciding that, well, actually, we're just going to let them do, not what they want. We're still going to say you have to use analgesia, but we're not going to say which one you need to use. and then assume that actually if the effect of the treatment is robust, that should not be influenced by the analgesic that you've used. But I think it probably needs to be done on a case-by-case basis. Like, what is actually important for the science? What is a factor that's going to be, confounding? And everyone knows that it's going to be confounding, and you probably need to think about that one. And factors where you're not really sure that, that might actually influence the result, but you're not sure, but maybe leave them up to the investigator. I think the crucial thing would be for this information to be reported properly so that you can actually make comparisons.
Yeah, I think reporting is we do agree. I mean, you should report as much as possible. But again, just asking you the general ones, but like within each technique and whatever, I mean, there's, we've had endless discussions, kind of like the same thing. And we're working on this. I mean, just actually about the general ones, but like the PCR experiments and the plasmid experiments and MTT experiments, each one have their own sets. And we're kind of like right now as reviewers trying to decide what's minimum, what you should enforce, and what just, okay, we recommend it, but if you don't want it, it's fine. Or we're not even recommending because we're being too picky, and it's not easy. Yeah. So
must be a hard balance to achieve.
Yeah. Anybody else there? Thiago, Silene, you comment about EDA. Any comments or questions that Nathalie should do? Anybody on Facebook?
No. Okay? No questions.
Okay, so yeah, I guess we're done. Thank you for hanging out. We've been having people come out. We still have a few people here. I'll be around for like another 5 minutes if you have any questions on the project to ask. Those of you that are part of the team. For those watching from outside via Facebook, etc., nice to meet you as well. Just as an announcement, we should be opening up our prediction markets for the Brazilian Reproducibility Initiative soon. So I mean, anybody with experience in experimental research can join in. We're going to like the final step of ethical approval, but it should probably be announced by the end of the month. Markets should run in November. So I mean, keep looking. Follow us in our website and social media, etc. You can actually sign up now and like just leave your email. We'll get in touch once those are ready. But I mean, that is something that anybody can take part in. And I think this is it for the moment. Thank you, Nathalie. Anything else from you, or?
No, nothing more for me. Thank you very much for having me. And yeah, I hope talk was useful. If anyone needs to contact me or send an email, my contact detail on the screen. I'm happy to share my slides as well if you want. I don't know if you want me to email it to you. Or that would be useful.
Yeah, you will. Actually, I have I'm working on my experimental design class for tomorrow, so that actually, if not for anybody else, probably I can make it bigger. I want to talk about EDA. Actually, can borrow a couple.
Sure, no problem.
Okay, so thank you a lot.
All right. Okay. Well, thank you. Bye.
Bye.